Okay, so now I'm going to start talking about the structure of a viron, its symmetry, and its shape. So let's start. So to start out, viruses come in many shapes and sizes, and a lot of viruses are way smaller than prokaryotic cells. So that's ranging from 0.02 to about 0.03. So let's go into the structure. So you have a capsomere, and that is going to be an individual protein molecules arranged in a precise and highly repetitive pattern around the nucleic acid making up the capsid. Some viruses only have one type of capsid protein, and some have more than one. So types that have one capsid protein, that's going to be an example like tobacco mosaic virus. Capsids can also be put together through self-assembly or spontaneously. They may require a host cell folding assistance. And now we're going to talk about the symmetry. So there's two kinds of capsomere arrangements corresponding. You have two primary. It's going to be your rod and your spherical, and that's like the two primary shapes of the viruses. So there's also different types of symmetry. So you have helical symmetry, and this is going to be a rod shaped, and it's determined by length of the nucleic acid. And an example would be the tobacco mosaic viruses, or TMV. So you have helicase, which is rod shaped, determined by nucleic acid. Then you have isosacchahedral symmetry, and this is a spherical shape. And these are going to be 20 triangular faces, 5, 3, or 2 identical segments. And these are most efficient arrangements of subunits in a closed shell, and this requires the fewest capsomeres. An example of this would be the human papillomavirus. Then you have a complex symmetry, which is a combo, and it's both elements. So it's cylindrical and spherical are present in both. Now we're going to move on to an enveloped virus. And these have lipoprotein membranes surrounding the neocapsid. Most use the outer surface proteins to attach an effect. So an example of this would be the Ebola virus. Relatively few enveloped plant or bacterial viruses because of cell walls surrounding cell membrane. Mostly present in animal cell viruses like the flu. And the entire viron enters an animal cell during infection. And then envelope viruses exit way more easily than they enter. And now I'm going to talk about some of the enzymes that are found inside of a viron. So you have a lysozyme, and this will make a hole in the bacterial cell wall to allow DNA or RNA to enter. This also lyses the bacterial cell to release new virons. Now we're going to go into the uramidases, and this is like the influenza. And these destroy glycoproteins and glycolipids. And they also allows for the liberation of viruses from a cell. And another type of enzyme found inside of a viron is the nucleic acid polymerases. And that's your RNA replicases and your reverse transcriptase. So your RNA replicase, it's going to be your RNA dependent and RNA polymerases. And then your reverse transcriptase is going to be your RNA dependent DNA polymerase in retroviruses, basically from RNA to DNA. Now I'm going to talk about plating of viruses. This is going to cover culturing it, detecting it, and counting it. And we do a lot of this in lab. So bacterial viruses are easiest to grow on a lawn of agar with an inculated space with a virus on it. Animal and plant viruses cultivated in tissue cultures from animal organ and culture medium or hairy root, or aka transfer root cultures in medium liquid. a type of plant tissue culture that is only used to study plant metabolic processes. And that's what's growing the viruses. So how do you detect and count the viruses? This is the plaque assay, is what it's called. So a titer is a number of infectious virions per volume of liquid. And the plaque assay is plaques are clear zones of cell lysis that develop on lawns of bacterial host cells where successful viral infection occurs. So you're going to calculate the titer from the number of plaques, and you're going to be analogous to counting colonies, which are going to be circles of viral growth. And this is a similar process for animal viruses as well. Plants are much harder, so viruses must be purified and counted microscopically or through viral protein-specific methods. And then basically a summary of all of this. So the cell phage mixture is poured into a solidified nutrient agar plate. The mixture is left to solidify. And then the incubation allows for the bacterial growth and phase replication. So that is what I have to say for my topics.
This is what our map looks like now.
This is now what our map looks like. And Maddie is going to continue with the rest of chapter 5.